So in the last class we had discussed the basics of the genetic engineering, that what are the steps that are used in the genetic engineering. Now today I am going to discuss with you the construction of the recombinant DNA, means how this formation of the recombinant DNA take place. Now first of all you should be clear what is the meaning of this recombinant DNA. As the word itself indicates, recombinant means it is the DNA formed by combining two different types of DNA. That's why it is called recombinant DNA. Now for the formation of this recombinant DNA, obviously we will require something, and the major thing that is required for the formation of the recombinant DNA is the plasmid. Now plasmid majorly act as the vector. Now what is the meaning of vector? The most common example that you already know is the malarial parasite that is being transferred by the vector female Anopheles mosquito. So female Anopheles mosquito, suppose this is A, which is being infected with malaria. Now the female Anopheles mosquito will bite this person. It will take the plasmodium from this person, and that parasite, means you can say the plasmodium, will be transferred to the B point. It means here the female Anopheles mosquito that is acting as the vector for transmitting this plasmodium from A to B. So in the same way the plasmid, they also act as the vector. For the formation of recombinant DNA we need two types of DNA. One is the host DNA and second one is the alien DNA. Alien DNA, what is the meaning of alien DNA? Alien DNA is the gene of interest that we want to introduce in a host, that is known as the gene of interest, or you can say this is the alien DNA. Now firstly you should be clear that what is the meaning of plasmid. Now all of you know that if I am drawing this as the bacterial cell, this bacterial cell is having the major chromosomal DNA and it is also having small extra nuclear rings. These extra nuclear rings, these are known as plasmid, means this is the plasmid DNA. Now what is the definition of the plasmid? These are the autonomously replicating circular extra chromosomal DNA. As you can easily explain this word from this diagram, that this is the ring, but this is not the part of nuclear material. It is different from this nuclear material. That's why it is known as extra nuclear ring. This is known as extra nuclear ring. This is circular. Autonomously replicating means it is capable of replicating itself without the use of any other help. So this is the autonomously replicating circular extra chromosomal DNA. This plasmid is isolated. Suppose I am saying that in the first step we had isolated this DNA. So this is the plasmid DNA. Okay, this is the plasmid DNA. Now this plasmid DNA contains the genes responsible for providing antibiotic resistance to the bacteria. What does it mean? That if this plasmid DNA is acting as the vector, why it is acting as a vector? Because in this plasmid DNA we had introduced a piece of DNA. For example, this is the piece of DNA that we had introduced. This is the gene of interest. Now when this plasmid will multiply, at the same time this gene will also multiply. Suppose I am labeling it as the A. So the number of times this plasmid DNA will multiply, this piece of the gene will also multiply. So plasmid, how it is formed? Now the question is that how it will be formed. Now this is plasmid DNA plus it is carrying the gene of interest, so you will say it is the recombinant DNA. Fine. When the DNA is formed by combining two types of the DNA, that will be known as recombinant DNA. So in this, one is the plasmid DNA and second is the alien DNA. So by combining alien DNA and plasmid DNA, the two types of the DNA, one type of the DNA is formed, that is the recombinant DNA. So how it is formed? See. Plasmid DNA was cut with the restriction enzyme. This was the plasmid DNA. If I am cutting it like this, from here I had removed this segment. Now this is the gene of interest. Suppose this is the gene of interest. So this gene of interest, now it will be introduced here. So what will be this? This will be the recombinant DNA. This recombinant DNA is then transferred into the host such as E. coli. This is the recombinant DNA. Now in the next step, this is the E. coli bacteria. This recombinant DNA will be introduced in this kind of the bacteria. Why this is introduced in the bacteria? Because bacteria, they multiply at a very fast rate. So the number of times the bacteria will multiply, in the same way this recombinant DNA will also multiply. So this is the use of the recombinant DNA and the use of the plasmid as the vector. So plasmid will act in the same way as the female anopheles mosquito acted during the transmission of the malarial disease. This plasmid is also carrying this foreign piece of the DNA into another cell. Similarly, the female anopheles mosquito is also transferring the plasmodium pathogen from the infected person to the healthy person.